Is this working okay? So first I'd like to thank the organizing committee and Human Frontier Sciences more broadly for the opportunity not only to present this work, but also to be a part of this really fantastic conference and organization. I think this is by far and away my favorite conference that I attend, and I hope to keep coming to these for as long as Human Frontier Sciences lets me. So I'm going to present a vignette of some research that was part of a young investigator grant that obviously from my beard I had quite a few years ago. Before that I'm going to just give a brief introduction on the biology of the system that we're working in. So as Barbara said, this is a really interesting organism. So we've got this bacteria Midichloria. If you know your Star Wars trivia, Midichloria gives you the Force. I don't think it does in this case, but we'll see. So in this context, Midichloria is a bacteria that's sort of related to Rickettsia, it's a pathogen, intercellular pathogen, and Wolbachia, which is an intercellular endosymbiont. It sits in the same sort of superfamily. And as Barbara said, there's this really interesting endobiotic interaction that's happening in this system, which as far as we know is unique in biology, which is that this bacteria has evolved to invade the intermembranal space between the inner and outer membrane of the mitochondria. And as far as we know, it's the only thing that does this. Interestingly enough, within this genus, there are species that engage in this behavior, and then there are species that are wholly cytoplasmic. And so that kind of set the scene for trying to understand the biology of these systems. We know that when they're inside the mitochondria, they appear to replicate, and they appear to degrade the mitochondrial matrix to some extent. We don't know whether it's pathogenic or symbiotic. I'll talk a little bit about that. And although it degrades the mitochondrial matrix, it doesn't seem to trigger cell death. So there's some interesting questions there too. So what do we know about this system? So some of the things, quick crash course in the biology of ticks. So ticks feed on blood in between molts during larval stages. And they also particularly feed the females for an extended period of time. And that large blood meal is what gives them the energy to produce a substantial amount of eggs. Midichloria amplifies in timing with these blood meals, and there's some indication, and one of the things I'll talk about a bit later in the presentation, some indication that perhaps one of the roles that Midichloria is playing in an endosymbiotic interaction with the tick is by helping it to produce endogenous heme alongside the heme that it's able to get from the host, because ticks lack some key precursors and cofactors that are needed to do endogenous heme synthesis. Okay, so it amplifies during blood meals. It's vertically transmitted. It's particularly abundant in females in the ovarian tissue and vertically transmitted at the end of the blood meal to the embryos. And that seems to be one of the ways that it transmits. It's hugely prevalent. You can, in principle, purge Midichloria from the ticks by treating them with an antibiotic like doxycycline. That's problematic because that has its own effects on the mitochondria itself. However, there's a nice natural experimental system that we can work in, which is that the European tick, Ixodes ricinus, has a Midichloria species that engages in intermitochondrial tropism. The related Midichloria found in the Australian tick, Ixodes holocyclus, is only cytoplasmic. And then there's a lab-reared strain of Ixodes ricinus called the Neuchâtel line from Switzerland that has been kept in the lab for many, many years, and it spontaneously eliminated Midichloria. So we had this natural system that we could kind of compare the effects between. What do we know about it? Well, from observations of that Neuchâtel line, in the absence of Midichloria, and this is kind of recapitulated when you do antibiotic treatment and purge Midichloria through artificial means. They don't feed for as long. Ticks can feed as females. They tend to feed for a couple of weeks, and they seem to drop off much more quickly. They have lower growth rate, lower fecundity. That could be consequence of the lower feeding time because, as I said, those two things are linked. And so we have these questions about the biology. Okay, so our study came in trying to understand this system better, and we had a number of really key questions that I imagine many of you are asking, which is how does it infect? Does it infect the mitochondria? There's a cytoplasmic population, there's an intramitochondrial population. Can they move from the cytoplasm to the mitochondria? If they do, how do they do it? What's happening? How does that affect the mitochondria? How come they can inhibit or seem to degrade the mitochondrial matrix but don't cause cell death? All these things. Are they pathogenic? Are they symbiotic? What is the nature of that interaction? What are they doing? Both at the ones that engage in intermitochondrial tropism or the ones that don't. And so then some of the challenges that we faced, well, there was as many challenges as there were questions. You can't grow these in the lab. You can, in principle, generate tick cell lines. There are a couple of different examples of tick cell lines, including ovarian cells, that you can have immortalized. And one of the sort of work modules for the program, which was done by colleagues at the Pasteur Institute, was to try and see if they could actually make in vitro infection models for these so we could study them. They're not culturable in any other ways. there's few genomic resources for these. There was one genome for Midichloria at the start of the study, and then we contributed. I'll talk a little bit about that in a few slides to try and bring that further along. And even working on ticks are a pain. Tick genomes are quite large. They're around about 2 gigabase, and they're really difficult to sequence. And that becomes problematic when it comes to looking at some of the tools we use, like RNA sequencing, to try and evaluate the effects of these organisms on the ticks, because you can't really do, or you couldn't really do, high-quality RNA-seq studies, because everyone would have to assemble their own de novo transcriptomes, and those are not particularly useful. And overall, the system was just really understudied. So I'm going to talk about a couple of vignettes of research, and then I'll come back to kind of overall program, but I won't talk about everything that was done in the project for the sake of time. So one of the studies that we were contributing to was providing genomic material from Ixodes holocyclis, the Australian paralysis tick that our colleagues at University of Pavia then isolated and assembled the Midichloria mitochondriae genome from. And this is the organisms basically classified as the same species or very, very closely related. And that allowed us to look at genomic markers that differentiated a species that only invades the cytoplasm versus one that's able to invade the mitochondria. And there's a couple other species where we could do this as well, and this was published just recently. And oftentimes I've done a lot of comparative genomic studies, and oftentimes they can be unsatisfying. You wind up with, you know, a couple of hypothetical proteins and some other things, and you squint and you hem and haw, and you go, oh, maybe that's doing something, but it wasn't the case in this case. It was really quite nice. So one of the things that was different between, so in red text, or at the bottom there, are 2 of the species that don't get engaged in intermitochondrial tropism, and the other ones do. And one of the really stark differences between them was the ones that engage in intermitochondrial tropism have a type 4 secretion system analogous to the types of systems we heard yesterday associated with Legionella, that are, again, used often by Rickettsia and other sorts of bacteria for invasion, and then flagellar proteins. And I paid attention to the talk yesterday from Matthew Baker, and I looked last night, and we definitely do have fly C there, as well as some mutagens. So it would seem like it's motile. So we have the organism that is in the mitochondria, has the machinery that are required to make flagella for motility, and in what appears to be an invasion system, which to me seems like a pretty strong piece of evidence that they must be able to invade the mitochondria, but you know, we'll see. And the other bits and pieces they have that the cytoplasmic species don't is a number of biosynthetic genes that are associated with heme biosynthesis and with production of biotin and folic acid, which are associated with heme synthesis. So it kind of fit with this idea that maybe they were inviting the mitochondria and they're helping with heme production. That was pretty cool. Okay, so then what we wanted to do was to use RNA sequencing to look at ticks across their blood meal when mitochondria or Midichloria are amplifying, and then see if we could understand what was happening to the tick tissues and also what Midichloria were doing. And so we undertook some RNA sequencing of a few different species. I'm going to focus on the data set that we have analyzed most comprehensively, which is the Ixodes ricinus system, where the Midichloria is engaging in mitochondrial tropism. One of the challenges we face is, although there is quite a lot of Midichloria in these tissues, when we did bulk RNA sequencing, there just isn't enough Midichloria there to get a good transcriptome to be able to do quantitative differential transcriptomics. However, because we now had the genomes for these organisms, we could work with Twist Biosciences, and we could get them to make a large comprehensive panel of exome capture baits, and we could use that to pull the Midichloria transcripts out of the ovarian tissues, and then we could get a nice signal. And suddenly now we could do quantitative transcriptomics, and we could look at what was happening. We saw lots of differential transcription across feeding, particularly in the mid and late stages, and then we could start to look at what the biology was telling us. Unfortunately, one missing piece in this data set was, even with exome capture, there just isn't enough Midichloria in ovarian tissue before feeding to get a good signal. So one of the things we saw, which was really exciting, was in our early feeding phase, we see that's when we see the most transcriptional activity of these flagellar systems and a lot of the type 4 secretion systems, which in my mind's eye suggests that once you start blood feeding, the Midichloria begin invading the mitochondria. I can't prove that. So one of the things we're doing next is to do quantitative RT-qPCRs on a subset of these marker genes, so we can look in the unfed tissues to see if that is the case, that we see a spike just in feeding and then it drops off. But nevertheless, there's some interesting data there that suggests that's happening. Though interestingly, one element that I'll call your attention to quickly is part of our secretion system that comes up late in feeding. One of the only things that comes up late in the feeding stage in Midichloria seems to be really interesting, and we'll come back to what I think might be happening there, but it's of course speculative. Once we move from the early to mid-feeding stage, the Midichloria transcriptome switches from this activation of these flagellar systems and type 4 secretion systems to upregulating a lot of pathways associated with ATP synthesis. A lot of these cofactors I mentioned are associated with heme biosynthesis get upregulated, and its replication systems, including the FTZ ring system, as well as some genes associated with cell wall synthesis. So this would indicate that the organisms are replicating, and they're starting to generate energy, and they're starting to engage potentially in this symbiotic reaction with the mitochondria. Sorry, I just said this. So these are some of the other different functional groups. Sorry to make you squint at the different colors, but one of the things I'll call your attention to down here are some of the cofactors that are associated with heme synthesis that are up in early and mid feed. And then the other part is, as I said, when we get into late feeding, a lot of this just drops right off. So when we look at late feeding, probably the major observation is that most transcriptional activity in Midichloria seems to drop right down, and I presume this is the organism going into sort of semi-quiescent phase as it prepares for this vertical transmission into the embryo. There's two things that buck the trend on this that are interesting. One of them is transposases. Transposases get highly upregulated at this point. It's really speculative as to what they're doing. The only thing we could say is that maybe they're associated with a lateral transfer from the bacteria to the host. Wolbachia does this, and there's some evidence that some Midichloria chromatin has left the Midichloria genome and gone into the tick genome, so perhaps there's something happening. Why do they do that? Is that what's happening? I have absolutely no idea. The other major group, well not group, the other sort of rest of the genes that are upregulated at that late stage are all hypothetical proteins. So we played around with the precursor to alpha-fold ITASR to try and get an understanding of what these might be doing, but it wasn't really conclusive. I'd like to come back to that with alpha-fold and see if we can get a bit of an understanding of what these things are. Beyond that, as I said, in late feeding, you just see dark for the most part. Everything getting transcription shut down. And this is replicated across the rest of the transcriptome. These are just some of the functional interesting aspects that I pull out. So it seems to be shutting down, except for these exceptions, with the lone exception being these, what would appear to be part of these type 4 secretion systems. And I wonder if that has something to do with preparing a little niche for itself while it waits for transmission, but it's speculation, of course. Okay, so the other part was what's it doing to the tick. Now we have the transcriptomic data across blood feeding in the ovarian tissue, but of course we can't separate out the massive changes that happen in the ovarian tissue with blood feeding and embryogenesis and everything else from what's happening that's associated with Midichloria itself. And so one of the other parts that we started to look at was what was happening between Ixodes ricinus. We're going to do a similar follow-up RNA-seq study in Ixodes holocyclus. That's the one where the Midichloria is exclusively cytoplasmic. And then we have this new Chattel line as well that we're going to do some RNA-seq work on. Before that, one of the co-investigators on the grant looked at the difference between this, the mitochondrial networks of Ixodes ricinus that has Midichloria and the lab strain that's lost it. And some of the things they found in, this is done with FibSem, was really quite interesting. In the wild type, where you have intermitochondrial tropism, and in this case, the Midichloria were in about 50% of the mitochondria within the cell. They have this really interesting morphology. When you look at the ones that's lost it, they have these long mitochondrial networks, these chains connecting up the mitochondria. But when Midichloria is present, the mitochondria are these little globs and sort of semi-discrete or spherical balls that aren't really heavily connected with each other, which is really quite interesting and quite a striking sort of contrast. And you can see this in greater detail here. So this is the Midichloria from the new Chattel line that's lost the symbiont. And then these are the mitochondria from those that haven't, and the sort of lighter green are the mitochondria. You can see that in some cases it can be quite a substantial amount of the biomass of the cell. So we did our transcriptional analyses of Midichloria in the new Chattel versus the Ixodes ricinus wild type line. They were just in the early stages of analyzing these data sets, but one of the things we see functionally is early on we see upregulation in the new Chattel, or sorry, in the wild type with genes associated with heme synthesis compared to the new Chattel line where Midichloria is not present. We see upregulation of some components of membrane integrity in the mitochondrial membrane integrity in the wild type. And then we see upregulation of these two NF-kappa beta and junk. You can speculate a million different ways on what NF-kappa beta and junk do, because as far as I know, they do just about everything. But at least in the context of mitochondria, they're important in this regulation of the balance between cell death and cell survival. And that might be interesting to think about in the context of Midichloria. As I mentioned, it's degrading the mitochondrial matrix, not causing cell death. We looked at the Midichloria genome, and there are a couple of genes in its genome that look kind of like they might, if you really squint, be structural homologs of BCL2, which is an important eukaryotic factor in inhibiting cell death. But it's, if you know BCL2, it's not the most complex protein domain. You could kind of fold a lot of things, so it's not really completely conclusive yet. Okay, I'll finish with a bit of hand waving of what I think is happening. So this is the fun story to finish my talk that I can't yet prove, but I think is happening. I think from our data, it seems pretty compelling. I find it impossible to believe that an organism has the complex flagellar motor machinery and just happens to be the version that invades or that's inside the mitochondria and isn't capable, it's not there to help it invade and move between, because what do you need a motility system for if you're sitting inside a membrane space in a mitochondria? So I think it does invade mitochondria. I think it does move from the cytoplasm to mitochondria. I think it is an endosymbiont. I think it is exchanging energy for providing cofactors for heme synthesis, among other things. And I think that probably that motility may even be a basal characteristic, because one of the things which I forgot to point out with the flagellar proteins is that those flagellar proteins that are present in the intermitochondrial species of Midichloria, we see pseudogenized versions of them in the non-mitochondrial, the cytoplasmic only kinds. So it seems like it's lost it through pseudogenization. So that's interesting. Okay, I'll finish here. So what have we done? So I talked about the sort of vignette around the RNA-seq work, but one of the challenging things of working with this system was before we could even do this work, we had to build the foundations to be able to just do some work on the organism. And so that was a lot of the focus, which I haven't talked about at all, around the Human Frontier Science Grant. My lab helped to develop genomics resources, but our colleagues did a lot of the imaging work, as I mentioned. In addition to that, they did work on trying to develop some polyclonal antibodies that they could fluorescently label the surfaces of the Midichloria to be able to track them in the cells. And there was an enormous amount of work trying to get them into cell lines that just wasn't successful. What I think this translates into is that we have something that is now a more conventionally fundable system we can work in. It's not completely ridiculous. We have some hypotheses that we can back up with preliminary data that we can now sort of pitch to a funding agency and work with. We learned a lot about the interactions, We learned a lot about the interactions of the organisms, as I mentioned, and then there's lots of stuff I'd like to do next. One of the other things that would be fantastic would be to produce antibodies against some of these flagellar proteins to see if we could track movement in the cells. I'd like to do some additional transcriptomic studies, including some of the stuff I've already described. And in the interest of time, I'll conclude there and acknowledge the work of the many co-investigators that helped with this study. And thank you very much.